introduction to some of our topics for this week. Our first topic is looking at the cell. And understanding that cells are the basic unit of life is a key concept. The human body is made up of tiny structural and functional pieces, which we call the cell. So when we talk about that the heart contracts to pump blood, it's the cells of the heart that are contracting to pump the blood. So we look at the functions of the human body by looking at the cells of the individual organs throughout the body and what their functions are. So they're very unique depending on the body system. So we look at, for example, here are these dark cells. These are called osteocytes, and these are found in our bones, and they create that hard bone tissue that gives us support and strength. These are nerve cells here in the yellow. Those are for conducting impulses and helping us respond to our environment. And these red cells in the top picture here are blood cells, and their job is to carry carbon dioxide and oxygen and keep our cells, all the cells of the body, provided with nutrients, or not nutrients, but gases such as oxygen, and picking up carbon dioxide to be exhaled. So we have a variety of cells in the body, and they all have a variety of functions. So when you look at cell size, the surface area to the volume has limits. For example, we can't have very large cells because it's just too much volume for that surface area. So there's an ideal amount of surface area to volume in which cells can function maximally. And when they get really big, sometimes those cells will divide, or sometimes those cells will die. Some cells have a limited lifespan, like our blood cells. They only live about 2 to 3 months. So when you look at cells, it's more ideal to have smaller clusters of cells because there's more surface area to volume for functioning and transporting and taking up materials than it would be to have one large cell. So this has a very large volume, if we look at it, compared to the surface area. For example, if I look at the surface area to the volume, there's not that great of a difference because the volume is so large. But if I take that cell and break it up into smaller cells, look at the surface area that I have. It goes from 96 to 384 if I break this down into smaller cells. The volume is the same, but the surface area is a lot larger. And that's ideal because, again, our cells are constantly absorbing and releasing materials to maintain homeostasis. So there's different types of cells. There are prokaryotic cells that don't have a nucleus or organelles, and those are not human cells. An example of those types of cells are bacteria. Bacteria are very simple cells. They do not have a nucleus. They have very limited abilities for functioning, but they can cause some pretty serious infections. But we also use them for making bread, for making foods, fermenting foods like soy sauce, different fermented things like sausage, and we'll get into that if you'd have to take microbiology. But eukaryotic cells, these are the cells that are found in the human body. They do have a nucleus and lots of organelles, and they can be very complex. So if you look at the structure of a typical cell, we'll see that it has a nucleus with a nuclear membrane. It has a small nucleolus. And the function of the nucleus and the nuclear membrane is to house the DNA. The DNA is found inside the nucleus in the form of chromatin, and that is all the genetic material we need to create another version of ourselves. So that important DNA is found inside the nucleus within the nuclear membrane. And the little structure inside of that is called the nucleolus. That's a special organelle, and its job is to make ribosomes. So other organelles we can see here, we have the mitochondria, we have the Golgi Apparatus, which is in charge of packaging and transporting different materials that are made inside the cell. And the mitochondria are for creating energy, ATP energy for the cell. And then we have the rough endoplasmic reticulum, which we'll always find has a membrane that's continuous with the nuclear membrane. Its job is to, it has ribosomes on its surface. That's what makes it the rough ER. And its job is to package proteins that are made on its surface and transport them inside the cell, and that might be taken up by the Golgi apparatus, further modified and packaged and released from the cell, or it might be used within the cell. We just don't know depending on that cell's functions. But this is the rough ER, this is the Golgi apparatus, this is the mitochondrion, This is a centriole. It kind of looks like a churro or like a Twizzler, little mini Twizzler, and its job is to create a spindle for cell division. When we look at mitosis, it creates that web-like protein structure for pulling chromosomes apart during cell division. And then this is the smooth ER. So this is a network of membrane that does not have ribosomes attached, and its job is to manufacture and transport fat molecules or lipids. So the rough ER manufactures or helps modify and transport proteins, and the smooth ER helps modify and transport lipids. So that's just just a general overview of the anatomy of the cell. We have these packaged little structures here called vesicles, and some of those contain digestive enzymes for breaking down unnecessary things inside the cell. We call those lysosomes. Those are important as well for just kind of keeping things cleaned up inside the cell. So all cells have an outer covering called the plasma membrane, and it's made up of 2 layers of phospholipids that makes it selectively permeable, which means only certain things can come and go from the cell. The cell is also filled with a semi-fluid substance. It's almost like a thin gel, and that's cytoplasm, and that helps, provide a fluid environment for different chemical reactions, and it suspends the organelles within the cell, has a very important function, gives the cell its shape. So we have this cytoplasm, and inside, floating in that cytoplasm are these organelles that we just mentioned. So if we look at the plasma membrane, you can see this blue ball with the little tails. This is a phospholipid molecule. So the phospholipid molecule there's 2 layers of phospholipids. You can see a layer here, and then you can see a layer here. So the ball side, the phosphate side of this molecule attracts water, and the tails that are oriented toward the inside repels water. So substances are attracted to the cell surface, but some of them can't pass through because of that fatty core. So fatty substances can pass through, but you know that water and oil don't mix, so things like sugars and salts that are attracted to the surface, anything that is water soluble, it can't dissolve in that membrane. It has to be transported across. So here's an example of a channel protein that will transport some of those water soluble substances across. So we have these phospholipid molecules, we have these large embedded proteins. Some of them have different functions. This one is serving as a channel. This one is attached to a carbohydrate, and this might be used for cell recognition and communication. These carbohydrate molecules are kind of the identifying structure on the outside of a cell. Just like your face identifies you as who you are, the pattern of carbohydrates across the outer surface of our cells helps other cells recognize one another. So this is called a glycoprotein because it's a sugar attached to a protein. And if that sugar is attached directly to a phospholipid, we call it a glycolipid. So this carbohydrate attached just to a lipid molecule, this phospholipid, that would be a glycolipid. So the proteins have many different functions in the cell. Like I said, they can serve as channels. Some of them serve as enzymes that help convert chemicals to one another in an enzymatic chemical reaction. And then we have these carbohydrates, which are for cell recognition and communication. We have the phospholipids that just build up the structure of the membrane. It's kind of like the brick and mortar, allowing certain things to pass through it. And then we have this purple structure. This is a molecule. This is cholesterol. Cholesterol helps stiffen the membrane a little bit because this is an oily substance, and the whole membrane would fall apart if we had just phospholipids. So having some cholesterol embedded among those phospholipids helps stiffen the membrane and keep it intact. And then we have these microtubules here that are kind of just on the inside, providing a little structure inside the cell. So when we look at the plasma membrane, we have proteins that are kind of floating across the surface. We have cholesterol for support. And like I said, those glycolipids and glycoproteins are for identification. So when we look at the plasma membrane, we have these proteins that have holes. Those are called aquaporins, so water can flow through the membrane. But things that are able to dissolve freely through the membrane are the gases like oxygen and carbon dioxide, or other fatty substances like nicotine and alcohol. Those can pass freely through our cells. They don't need a protein transporter, but water does. Here's an example of an aquaporin, but here's something that's water soluble. It hits that fatty core and it can't pass through, so it needs a transporter to get it across. So the way things come across the membrane is by diffusion, where we have higher concentration of particles going to lower concentration. Osmosis is where we have water. It's only referring to the movement of water. So we have more water on one side moving to where there's less water on the other side. We have facilitated diffusion that doesn't require any energy from the cell, but it requires a protein to carry it across the membrane. So that would be for water-soluble substances that can't dissolve through the membrane. It needs a protein helper, but it doesn't require any energy, so things are still moving from that high to low concentration. Active transport does require energy, and that's when we want to move things from low concentration to higher concentration. So that we'll look at in just a minute. And then endocytosis and exocytosis, these are energy requiring processes where we're taking a membrane of the cell and either bringing it into the cell by wrapping a membrane around it, and that would be endocytosis, or we wrap a little membrane around something that's been produced by the cell. Say, for example, the rough ER or the Golgi Apparatus, and we release it from those organelles, and that's called exocytosis. So endo is bringing things inside, and exo is taking things and sending it outside. So when we look at diffusion, key thing to remember is we're going from higher concentration to lower concentration. So this is a natural process. It's passive, there's no energy required, and it's going to continue until things are equal. So if I look at this example here, the purple particle, we can see there's more of this purple particle on this side than there is on this side. So that purple particle, as long as it can diffuse across the membrane, it's going to move across until the particles are equal. So if I look over time, I'll see there's more purple on the left than there was in the beginning, and they're roughly about equal. So we have this movement of water and the particles back and forth until they're equal. So again, the movement of water we call osmosis, the movement of the purple particles we call diffusion. But regardless, there's no energy required, and they're moving from where there's higher concentration to where there's lesser concentration until they're equal. And that's what we see here on the right-hand side. We see equilibrium. There's equal amounts of water on both sides, and there's equal amounts of those purple particles. So osmosis, again, is the movement of water from higher water to lower water. If we have two substances that have equal amounts of water or two solutions, I should say, like inside the cell, if the amount of dissolved substances inside the cell is the same as the solution outside the cells don't take on or release any water, any net amount of water, and the cells are stable. Just like right now, your blood cells and your plasma are in an isotonic solution. Your plasma, the fluid part of your blood is isotonic, so your blood cells are happy and there's no swelling or shrinking of your red blood cells. But if I threw those red blood cells into pure water and not plasma, what would happen is your red blood cells would take on water because there's more water outside the cell than inside the cell, and that water is going to flow in, and it's going to cause them to burst. So the solution outside the red blood cells is called a hypotonic solution because it has more water than inside the red blood cells. So hypotonic means more dilute. Hypo means low, and tonic refers to dissolved things in that solution. So low solute, low salt, low sugar, whatever, low substances in a fluid is called a hypotonic solution. Now this is a hypertonic solution because the cells are losing water, so we have more water inside the cell compared to outside the cell, meaning that this blue solution here is a concentrated solution. Maybe it's really high in salt or high in sugar, and that causes these red blood cells to lose water because there's more water inside the cell than outside the cell. So this is a hypertonic solution here, and we can see the water level is going up a little bit because the water is leaving the red blood cells and entering into the solution. So Ideally, we want isotonic solutions, right, for our cells. We want our cells to be in a happy solution where there is no movement, big movements of water causing those cells to shrink or swell. So cells will shrink in a hypertonic solution, and that's again a concentrated solution like a high salt or high sugar water, and our cells will swell in a pure water solution. So looking at different parts of the cell, we talked about the nucleus contains that DNA, the genetic instructions for the cell, and there's a membrane system inside some of these organelles like the Golgi apparatus and the rough ER. So when we look at the nucleus, the DNA is in the form of chromatin, which is kind of just like a pile of spaghetti. And when that pile of spaghetti becomes very organized and tightly coiled, it forms little spring-like looking structures called chromosomes. And you've heard of chromosomes before. And a section of a chromosome that codes for a specific protein, we call that a gene. So genes are just regions of DNA that code for a protein. It's basically the recipe for making a protein in the body. So this DNA is found in the form of chromatin when it's loosely bound, and chromosomes when it's tightly coiled. And that's just our genetic information. And then we have fluid inside the nucleus to suspend this DNA. It's called nucleoplasm. And then we have that small round structure called the nucleolus. And its job is to produce ribosomes. And that ribosomes are necessary for protein synthesis. So here's that nucleolus. There's the chromatin. That's the DNA, that loosely bound genetic material. We have the nuclear membrane, and there's pores in the membrane for things to come and go from the nucleus pretty freely. We have this rough ER, which is the network of membranes attached to the nuclear membrane, and it has these ribosomes studded on its surface. So it's kind of like a beaded purse, these studded ribosomes. And the ribosomes make protein, and then those proteins are modified and packaged by the rough ER. And then we have the smooth ER, talked about that already. That's for lipids. So lipid molecules, fats, are manufactured and transported and packaged by the smooth ER. So different functioning, similar appearance, but remember the rough ER has those ribosomes on the surface. So the nuclear envelope surrounds the nucleus. It has those pores to allow things to come and go. And then, like I said, we have these ribosomes. Ribosomes are responsible for making proteins. And there's lots and lots of things that are made up of protein. For example, insulin is an important hormone for breaking down sugar in our body and allowing sugar to enter our cells, and that is a protein hormone. Your skin is made up of protein. Our muscle is made up of protein. So we need these ribosomes to make those proteins. So we find them on the surface of the rough ER, and we also find them floating freely in the cytoplasm of the cell. So we have free ribosomes and we have attached ribosomes, and their job again is to make protein. So again, this endomembrane system are those special organelles and structures that are membranes, which is the Golgi apparatus. The lysosomes are surrounded by a membrane. Those are those small little yellow structures, remember, that contain digestive enzymes. And any membrane structure in the cell is called a vesicle. So these are all examples of, I'll just go back here. Actually, we'll go forward. You can kind of see what these look like. So like I said, the rough ER has ribosomes on it to help make proteins. The smooth ER does not have ribosomes. Its job is to make fatty materials, which we call lipids. And the Golgi apparatus works with with the smooth and rough ER to modify those proteins and lipids and package them for use either inside or outside the cell. So the Golgi apparatus is kind of like that last receiving organelle to finalize these products for use in or outside the cell. So here's the rough ER, the brown is the smooth ER. The Golgi apparatus is going to take up either those lipids or those proteins made by the ERs, packages it, and sends it either outside the cell or keeps it inside the cell. And again, any substance wrapped with a membrane we call a vesicle. And if that vesicle contains digestive enzymes, which is a protein, we call it a lysosome. So a vesicle is kind of a generic term, where a lysosome is a more specific term saying that what's inside that vesicle is a digestive enzyme. So these are just small membranous sacs, just kind of a generic term, and then the enzymes are the lysosomes. So we have other structures that extend from the surface of the membrane, the cell membrane. Cilia are short, numerous little structures. Their job is to move mucus, like in the respiratory tract. And the flagella is a single whip-like structure, and its job is to move the cell itself. So the only cell in the human body that has a flagellum is the sperm, and its job is to carry that sperm up to find the egg, which is released from the ovary once a month in the female, and it hangs out in the fallopian tube until the sperm swims its way up and locates it.